Hello, my name is Jamie Rosas.
Hello, my name is Mohammed Hassan, and today we will be talking about hormonal control of the cell cycle. So, for the start, we'll be talking about the basic components of the cell cycle, which includes the G1, S phase, G2, and mitosis and cytokinesis. So, the cell increases in size during G1 phase, and then it copies its DNA during S phase, and the cell prepares to divide in G2 phase and finally divides in the mitosis. And there is a fun fact that, in contrast to animals, plants continuously generate new organs throughout their lifespan. This developmental plasticity requires a strict temporal and spatial control of the cell cycle. Now, I'll be talking a little bit about the biological significance and developmental process of the hormonal control of the cell cycle. So, plant hormones play a crucial role in regulating various aspects of the plant cell cycle, including cell division, differentiation, growth, and stress response. Plant hormone regulation is crucial for the proper functioning of the plant cell cycle, and the developmental process for plant hormone regulation in the cell cycle involves the initiation of hormone production, signal transduction, interaction between hormones, and feedback regulation to ensure proper cell division, differentiation, and growth. And then on the chart on the right side, you can see different plant hormones and how they impact germination, growth to maturity, flowering, fruit development, abscission, and seed dormancy.
So, like mentioned before, the cell cycle is influenced a lot by the hormones and their interactions, again, with their ratios. But actually, some molecules that promote transition from phases to phase would be these molecules that I'm going to mention, one of them being a cyclin-dependent kinase, which is also known as CDK, cyclin, and inhibitor of CDK, which is also known as ICKs. These all come in different families, so you can have different types of CDKs, different types of cyclins, and different types of ICKs. Again, the CDK is a family of kinases that affect usually a threonine or serine. Cyclins are also a big family that actually are, you can say, are the activating unit that helps activate a CDK. They actually form a complex, a CDK cyclin complex. And again, in the blue, you can actually see the CDK. And that CDK is going to be where the reaction happens, or it's also known as a catalytic subunit. And the cyclin is just the activating subunit. So the CDKs can be present in the area of transcription or where the cell cycle is going to basically do, but it's not active until it's actually bound into a complex with cyclin. As for the inhibitors, here I have just representation in red of what an inhibitor, nothing what it looks like. But basically, this is going to be what inhibits the CDK cycling complex from actually having any activity. So yeah, so moving on to the next slide, and we're going to talk about G1 phase in plants. So the G1 phase in plants is where the cell either continues through the cell cycle or starts differentiating. A key factor in differentiation is a retinoblastoma protein, which is here in red. And this retinoblastoma protein, also RB, basically helps in controlling the differentiation status of cells. So a deficiency in this molecule delays cell differentiation, whereas in excess induces expressions that lead to basically differentiation pathways in cells. And again, these are just molecules that play a key component in the first transition, which is the G1 phase to the S phase. We have CDKA in blue, and these are the hormones that help induce or promote that type of CDK, as well as cyclin D. We have a ICK, which again works as an inhibitor of the pathways or the transition from G1 phase to S phase. And this inhibitor is actually induced by ABA. And inhibited by auxin. And all these again work in tandem to help transitions through the cell cycles. We also have CDKD, which plays a key role in G1 to the S phase, as well as we have an E2F DP complex shown here, and I will mention that again in the next slide. So in the next slide, we're gonna actually talk about the mechanism behind the transition from G1 to the S phase. And here on the left, you can see this molecule. And basically, it's a combination of CDKA, cyclin D, and ICK. And at this stage, this is inactive due to two reasons. One of them being because of the ICK being binded to this complex, the CDK cyclin D complex, but also because of the catalytic site lacks a phosphorylation on the threonine 160, and that is key in actually making it active. So here, if we move down the pathway, the ICK is actually going to come off, and there still needs to be a lot of research being done on how that actually comes, if what helps promote that being taken off. But again, it could be the concentration of the ICKs present in that actually has to do with the with that whole subject. But that's going to come off, as well as CDKD is going to help phosphorylate that threonine 160 on the CDKA. And so once that phosphorylation happens, this becomes an active CDK cycling complex. So here we have again that complex in an active form, and that comped with the RB. At this point, the RB is binded onto the E2F, our DP complex, which is these two are transcriptional factors that help turn on, basically promote transcription of specific genes that help promote or progress into the S phase. And so what the CDK cyclin complex is going to do is actually going to phosphorylate the RB molecule. And in doing so, it actually unbinds the two molecules that were once held together. So we have a liberated E2F and DP molecule or complex. And so basically, that's going to, again, turn on transcription. And here we have a more like professional description of what's happening. Here we have a transcription off because you have the RB protein binded and basically just not working. And once we have those CDK cyclin complex phosphorylate the RB, you basically have an or you basically turn transcription on. So moving on, just again in the S phase, you have that synthesis of a complete copy of a DNA in its nucleus, and that's being done by the E2F DP complex that promotes transcription necessary for progress into the S phase. With that whole DNA replication in the nucleus, we also have a new molecule, which is the WEE1, which is the gene is expressed by the induction of, or the is induced in the response to the DNA replication checkpoint. So that's going to come into play in the next slide. So in the G2 phase in plants, again, we have just the cells replenishing energy storage and synthesizing proteins and organelles necessary for the next phase, which is mitosis. And the cell can also grow additionally. And I'm just going to mention the key factors in the G2 transition to M phase, and that's going to be CDK A and B, cyclin A and B, a molecule known as CAK, which is a combination of CDKD as well as CDKF. So that right there is CAK. So just again, a combination of two CDKs. Yes, two CDKs. And again, our molecule that we talked about in the last slide was this WEE1. And then here again, we have kind of what promotes them, what hormones promote that. So we have auxin, gibberellin, kind of are the ones that promote both CDK and cyclin. Moving on to the next slide, we're going to have the mechanism from the transition from G2 to M phase. And here on the left, we have, sorry about that. We have an inactive form of the molecule, which is basically, you can see that on the CDK, you have a phosphorylated T14 and Y15, and you don't have the phosphorylation on the T160. And that's basically going to be an inactive form of this CDK cyclin complex. So there's going to be two steps needed to basically make it into an active form. The first one being done by WEE1, which is actually going to dephosphorylate this the T14 Y15. And so in the next, you can see that both are non-phosphorylated. So that's basically an intermediate. And then we're going to have the CAK molecule phosphorylate the T160. And then once you phosphorylate the T160, you basically make this an active molecule. So moving on, again, this again helps from G2 to M phase. And then once you have you're in the M phase, you're basically dividing into two identical daughter cells. But a key component into exiting the M phase and going back into the G1 phase is actually this process known that degrades the cyclin. And the degradation happens by anaphase promoting complex, so APC, which is this just illustrated in red. So what this will do, it will actually target the A, B type cyclins and basically have them degraded using a E3 ubiquitinate ligase. And so you can just see here, you still keep the CDK A and B, but you're degrading, we're getting rid of the cyclins. And then that's going to promote the movement from N phase to G1 phase. So yeah. And then in the next slide, I'm going to quickly just talk about one hormone in specific, and it's going to be auxin in the roots. And basically here we can see how multiple hormones interact to basically have the cell elongation, cell differentiation, cell proliferation, as well as other factors. But specifically, I'm going to talk about auxin real quick. So auxin, there are auxin responsive genes that can be transcribed that help in the regulation and of other molecules responsive to auxin. And what we're going to say is you have auxin present, or basically, let's say you have AUXIAA transcription repressors present if auxin isn't present. So those are actually binded to this ARF molecule, which is a transcriptional factor that actually, if not binded to the transcriptional repressors, bind to the promoter and induce transcription of those auxin genes. So the pathway that is simple is if auxin is present, you have the promotion of TIR1. And this TIR1 is going to inhibit AUXIAA transcriptional repressors. And if that is inhibited, you have a promotion of ARF transcriptional factors, which create that biological response. So that is just a simple pathway that kind of helps explain how just the cell cycle is complicated, that many factors play key roles, but again, it's crucial for cell differentiation for many aspects of that are involved in cell cycle. So yeah, that's basically our presentation in what we try to do in 10 minutes. Here's our references, and yeah, thank you again.
Thank you.